DNA is the genetic code bound within chromosomes inside the nucleus, or the center, of each cell. It contains directions for making proteins, which is done in ribosomes outside the nucleus. But DNA can't leave the nucleus, so it produces a copy of itself called messenger RNA, or mRNA, which can carry the blueprint for making proteins outside the nucleus. Proteins are key to the structure and function of each cell, such as supporting the cell's outer barrier or membrane, executing chemical reactions, and clearing harmful substances. Genetic disorders occur when there are one or more changes in the sequence of specific units of DNA known as genes. These changes may disrupt the production of functional protein, leading to health problems. For example, there may be a breakdown in regulating what goes into and out of cells because important membrane structures aren't working, or toxic substances may build up within cells because there aren't enough functioning proteins to break them down efficiently. mRNA therapy is being studied to treat many different health conditions, including some rare genetic disorders caused by protein deficiencies. Researchers believe mRNA therapy can provide the blueprints that cells need to make properly functioning protein, thereby reducing symptoms, modifying the course of disease, and improving quality of life. This approach uses mRNA as a protein replacement therapy. As a protein replacement therapy, the specific mRNA unit needed to make the missing or dysfunctional protein is administered. Therapeutic mRNA is usually transported to cells by lipid nanoparticles, which are tiny fat bubbles that surround and protect mRNA. It may be given as an infusion into the bloodstream. mRNA therapy needs to be given at regular intervals because the therapeutic mRNA is broken down in the body. mRNA therapy is being developed for several conditions, including lysosomal storage disorders, cystic fibrosis, or CF, and familial hypercholesterolemia, or FH. Lysosomal storage disorders are a group of genetic conditions that are a result of problems in the structure or function of the lysosome, or the cell's waste processing center. Most lysosomal storage disorders are caused by genetic changes that result in reduced production of one of multiple lysosomal enzymes. Lysosomal enzymes help cells get rid of waste products. In Gaucher disease, for example, the enzyme glucocerebrosidase does not work well, and fatty material that is typically broken down by this enzyme builds up in the liver, spleen, and bone marrow. mRNA therapy for Gaucher disease aims to provide the blueprint to manufacture glucocerebrosidase, so the cells can once again get rid of excess waste products and reduce symptoms of the disease. CF is caused by changes in the cystic fibrosis transmembrane conductance regulator, or CFTR gene, resulting in loss of CFTR protein function. The lack of functional CFTR protein affects how cell membranes control electrolyte and water flow. When CFTR does not work properly, too much mucus is produced, leading to chronic wet cough, risk of lung infections, and chronic lung disease. The pancreas, liver, intestines, and sweat glands may be affected, as can growth and male fertility. The goal of mRNA therapy for CF is to increase normal CFTR protein production and improve function of the lungs, as well as potentially other affected organs. FH is a group of genetic disorders that result in very high levels of LDL, or bad cholesterol. The low-density lipoprotein receptor, or LDLR protein, binds to the LDL and removes it from the blood. In the most common form of FH, there is low LDLR activity due to one or more changes in the LDLR gene, leading to cholesterol buildup on artery walls and increased risk of early heart disease, disease of the arteries in the arms and legs, and potentially stroke. mRNA therapy for FH may be used to restore LDLR levels and reduce the risk of heart disease. These are just a few examples of where mRNA therapy is showing promise. By improving cell function and restoring protein activity, mRNA therapy may also improve quality and length of life for individuals with these chronic conditions. Advantages of mRNA therapy may include mRNA is not incorporated into a person's DNA, so it is not expected to negatively impact the production or function of other proteins. mRNA can be customized to each genetic disorder. mRNA can be modified to reduce the risks of immune system reactions, thereby limiting side effects. And the nanoparticle delivery system can be programmed to target specific organs and to protect therapeutic mRNA from being broken down before it reaches its destination. mRNA therapy also presents with challenges and potential side effects. It is a lifelong treatment that requires repeated doses at regular intervals. Side effects can vary based on the therapy received. Some side effects may include reactions at the needle site, infections, malfunction of implanted devices, such as a port used to make repeated infusions easier, flu-like symptoms such as headache, nausea, and fever, inflammation of the heart and liver, and allergic reactions. As a quick recap, mRNA is currently being investigated as a protein replacement therapy in many genetic conditions. mRNA therapy may be used to provide the blueprint needed to make missing or dysfunctional proteins. mRNA therapy typically requires repeated doses and may cause side effects. Researchers believe that mRNA therapy represents an exciting future treatment strategy to possibly improve symptoms, change the course of disease, and improve quality and length of life.